Welcome to the third video in the unit for prokaryotic cell biology. In this video, we're going to talk about the various filamentous external structures that protrude from different bacterial cells. Let's go ahead and get started.
The external structures of bacteria allow the cells to interact with the environment in some way, and these interactions can help expand the cell's habitat or allow them to survive better. The two main sets of structures we will discuss are the Puli and Fimbre and the Flagella. Fimbrae and pili are two different types of filamentous structures that can be found extending from the cell envelope of the bacterial cell. They are both involved in bacterial cell attachment. The main difference between them is their structure. Fimbrae are typically short, stiff filaments that are very numerous over the cell, while pili are usually longer, thicker, and more flexible. Besides attachment, there is also a certain classification of pili that function in the transfer of genetic material between cells, a process called conjugation that we'll discuss later in the course. Attachment is important for many bacterial cells, so they're not thrown about their environment. For example, it might be more beneficial for a cell to attach to a rock in a flowing stream than tumble along in the water. Attachment is a key step for any pathogen because in order to invade the human body, the cells must first attach to our own, or they'll be removed from the body. For example, a respiratory pathogen must attach to the lining of the lung epithelium or be expelled when you cough or sneeze. While some organisms want to attach to a surface, others control their destiny by being modal so they can swim throughout their environment. These species contain flagella, which propel them along. While some eukaryotic cells also contain flagella, think about the shape of a human sperm cell, for example, the bacterial flagella is very different both structurally and in its movement. Bacterial flagella are long filaments made out of subunit proteins called flagellum. They're attached to the cell through a hook structure that is bound to a ring that's able to spin in the membrane. This ring structure is able to spin both clockwise and counterclockwise, allowing the flagellum to move just like the propeller of a boat. So, for example, the flagellum would be moving in circles this way, and then it can switch and move in circles the other way. This is very different than a eukaryotic flagella, which just moves side to side in a wave-like motion. Flagella are very thin structures, so they cannot be seen in a light microscope unless you specifically stain for them. There is a flagella stain that will use a mordant, allowing the dye to build up on the structures until they are thick enough to be able to see in the light microscope. The presence or absence of flagella can begin to differentiate one bacterial species from another, but so can the arrangement of flagella on the cell surface. There are 4 flagellar arrangements: monotrichous, which have one single flagellum; amphitrichous, with one flagellum at each end of the cell; lophotrichous, which means it has multiple flagella but they're all clustered together at one end; and peritrichous, which are spread all over the cell. No matter the arrangement of the flagella, all bacteria swim using the flagella in the same way, called a run and tumble movement. When flagella are spinning in a clockwise direction, it causes the cell to tumble about in 3D space. When the flagella stop and switch to a counterclockwise motion, it causes the bacteria to run in a straight line. The bacteria are not swimming randomly around their environment. They have receptors that allow them to do something called chemotaxis, or moving in the direction based on a chemical. They will swim towards attractants, such as food, and away from repellents, such as toxins. The purpose of run and tumble is that they can ensure they're traveling in the correct direction. So, for example, there's a food source over here, and your bacteria with their flagella are down this way. That flagella might spin in a clockwise rotation, causing the bacteria to tumble about in 3-dimensional space. Eventually, its receptors will pick up the food, which is diffusing. We've got a high concentration here and a lower concentration as it gets away, and that will orient the bacteria in the proper direction, and the flagella will now switch to a counterclockwise rotation, which allows the bacteria to run in a straight line. It'll get to a certain point. The flagella will again switch to running in a clockwise direction, allowing the bacteria to tumble around. This is so that it can check and orient and make sure that it's still in the right location. Typically, bacteria do not go in a straight line. They might run this way for a little bit, Stop and tumble a few more, make sure they're oriented, and run this way until they eventually get to where they are trying to go.
Remember, we just discussed that bacterial flagella move like a boat propeller, where they spin in one direction and the other direction. This video is going to let us see that motion because the flagella actually got itself stuck to the microscope slide, and so now when it spins, instead of propelling the bacteria along, it's just spinning the bacteria in static space. So we're going to focus on the center bacteria here, and you'll be able to see as the flagella is moving, you can see how fast that cell spins itself. As we watch, we can also see other bacteria completing their runs through the liquid media, some of them going quite fast, others going a little bit more slowly. And as we watch, I really like this big guy as he comes mosey on through. But that should give you a sense of how bacteria move in their run and tumble movements, and hopefully now you have a better understanding of the various types of external filamentous structures that are found in prokaryotes.